Hello. Today I just want to go over this assignment with you real quick. And just show you a couple things about it. So the first thing I want you to do is watch this video in this link. What is mutations? Just like 2 minutes. The next thing is go to this page, mutate a DNA sequence. It'll take you to this here. This interactive we'll play with in a sec, and I'll show you how it works. The first thing though is that I want you to read. Actually, I'm going to read through this with you, and then you'll read through down here and look at these different kinds of mutations. But first, I want to go over this codons and reading frame thing. So you guys should know what codons are already because we've been working with that genetic code chart. So the sequence of 3 letters is a codon, and it gives us an amino acid, right? So let's read this real quick. So it says cells read a gene's DNA code as a series of 3-letter words called codons. Each codon codes for a specific amino acid. Genes also have a reading frame. Take this DNA sequence as an example. So here's the DNA sequence. And now it says you could divide it into 3 ways, each with a different reading frame. So depending on where we start reading this, we are going to read it in a different way, and that would give us a different protein every time. So if we started at the beginning with the G and read GCA, well, that GCA gives us a certain amino acid. TGC gives us a certain amino acid, and so on. However, if we shifted over, if we changed our reading frame, basically with which 3 we start with, now we read cat, and then we read GCT and GCG, and those give us different amino acids than this first sequence, and so on with the third one. Okay, so we start with ATG there, and that's going to be different from the first 2. So this is only one of these reading frames will code for a working protein. How can you tell which is the right one? It's a little like finding the right spacing for letters in a sentence. For example, here is the sentence: the sun was hot, but the old man did not get his hat. So we have to find the right spacing, and we have to start reading with the correct letter. Okay, so I want to give you another example of this. It says the fox ate the sad cat. So codons in mRNA are like words in a sentence, right? The mRNA is a message, and we have to break it down into pieces. We talked about this, but so this is how we read this. We start with the fox ate the sad. Okay, you go. So we read starting with the T. We read the sentence like that. But let's say we had a mutation. Okay, let's say that, for example, after the H, there was a mutation and we accidentally put another letter in here. What if we put another, let's put an H? I don't know, it doesn't matter. So now this would change our whole sentence. So look, it's going to be the THH, and then the E bumps over to the beginning of this one over here. So we have EFO, then we have XAT, then we start the next one, ETH, ESA, and then DCA. Oh, and then T. Okay, and so you can tell that we added a letter because we have one extra hanging T right here. So basically what we did by inserting this letter, we changed our reading frame, and now we are off by one letter the whole time, but that messes up everything. After the mutation, that messes up every single one of these, okay? The same thing would happen is if we deleted a letter from the original. So now we're going to have T, H, and then the F gets bumped over to the first part, and then O, X, A, T, E, T, H, E, S, A, D, C, A, T, and now you can tell that we deleted one because we're missing one right there, okay? So this whole idea of reading frame is basically just reading the correct 3 letters at a time, and if we insert one or we delete one, we're going to mess up that reading frame, okay? So that's what a reading frame is, and so in this thing here, I ask you to answer that question, and then down here, describe the different types of mutations. You're going to scroll down a little bit and go here, okay? The next thing that it's going to ask you to do is use the interactive at the top of the page to answer these questions. So this guy is cool because it shows exactly what I just showed you. So here is the DNA, right? Here is the RNA, here's the protein. So if we were to add a letter or delete a letter, that's going to mess up the different amino acid sequence, right? So for example, let's say I take this G, and if I click it, I can do different things. I can change it to one of these different letters, like let's say I change the G to a T. That changes the mRNA, and then it highlights the protein that it changed right here. It changed it to SER from whatever it was before, okay? We could also delete the letter, and when we delete it, just like we saw with those sentences, everything shifted over to the left, and now it highlighted all of the amino acids that got changed because we are now in the wrong reading frame after that mutation, right? So over here in the corner is a reset button. It's kind of hard to see, but yeah, that's basically what we can do. We can add one too by clicking one of these little arrows. So if we add one right here, this is supposed to be, what does this highlight? Alanine. So if we add, you have to click out in the box. If we add one right there, bam, that alanine changed to ASP, and then all of them to the right changed after because we changed the reading frame after that mutation. Okay, so it asks you to do certain things in here. It asks you to like just go to this amino. You could also click on one, and it highlights. If you click it, it highlights the DNA and the RNA that you need to change. So you can change, any of these things there. Okay, so let me know if you have any questions, but that is pretty much the gist of it. Thanks, guys.